Hello, everyone. Good day. My name is Byron Cheatham, and I'm Vice President at CytoViva and a principal in the company. And today, I'm going to be joined by Dr. Phoebe Stapleton. Dr. Stapleton is the Associate Professor of Pharmacology and Toxicology at Rutgers University in New Jersey. And today, we're going to be sharing a lot of details regarding nano and microscale plastics imaging and analysis. CytoViva provides enhanced Darkfield and hyperspectral microscopy that's utilized extensively in research of nano microscale plastics. And Dr. Stapleton and her team are extensive users of the technology, also are involved deeply in a wide range of studies related to the toxicological effects of micro and nanoscale plastics. And she's going to spend a good bit of time talking about the scope and scale of her research, not just what includes the CytoViva instrument, but also just more of a general overview of her work in that regard. And we're pleased to have her join us today. We're pleased to have you join us as well. So with that, I want to get started a little bit here and talk about the agenda. We're going to very briefly discuss how dark-field hyperspectral microscopy works, a little bit about CytoViva, the company, and some emphasis on nanoplastics applications. I'm going to go through a couple of very brief application examples. Then I'm going to turn the webinar over to Dr. Stapleton, and she's going to give about a 35 to 40-minute overview of her research in nanoplastics. And her research focuses a lot on non-traditional models of exposure to nanoscale particulates and plastics in particular, and exposures incorporating reproductive toxicology. Her studies focus on exposures during pregnancy, leading to the development of a host of gestational environment identified through microvascular evaluations of the mother. So it's really interesting, and her work has gotten a lot of press lately. It's very interesting work, and I think you're going to like it. After we finish this today, we'll have questions and answers. So with that, just to briefly let you know a little bit about CytoViva. CytoViva is a 20-year-old imaging company that was developed and designed to work in the nanoscale space. The company has a presence around the world. Approximately 400 research labs around the world utilize CytoViva technology. And these are principally research labs that are involved in some sort of engineered nanoscale research or nanoscale particulate exposure work, which is a focus that we're looking at here today, looking at a wide range of polymers and plastics. The CytoViva instrument and technology is referenced in well over 1,800 scientific publications. So there's a lot of use and experience with the technology. I very briefly want to just sort of describe at a high level what this technology does and what it does in the context of nano and microplastics work. The first thing you see an image here on the right, and this is an H&E tissue image. We're looking at these white particulates, which are nano and microscale plastics that have internalized in this tissue. And here we see a spectral response, and that's a spectral response of one of these white particulates, which happen to be the polystyrene plastics, which are there. So with this instrument, you have the ability to optically observe, without any special sample preparation, nanoscale samples. And you can observe those in a wide range of environments. They can be observed in solutions. They can be observed in vitro and cell culture. They can be observed in ex vivo tissue, as we're looking at here. This happens to be stained ex vivo tissue, but also a wide range of material matrices, thin films, and that sort of thing. And then, as you can see by the spectral response of a specific pixel of data for the plastics that are here in this tissue, you have the ability to spectrally identify and characterize these nanoscale samples, and then you can Based on that analysis, you can do spectral mapping of these nanoscale samples in a wide range of environments. In a nutshell, it's designed for you to be able to see and confirm the presence of nanoscale materials in a wide range of environments. This just gives you a sense of the footprint of the system, and you see it's a basic research grade optical microscope. And what's unique is it are the optics and the spectroscopy that's added to this research scale optical microscope to allow it the ability to be able to observe at the nanoscale and record optical spectral data in every pixel of a hyperspectral image. We'll explain what those are, but these are the two key elements on the microscope that allow for this nanoscale imaging and analysis. The first thing I want to do is talk about the patented enhanced darkfield microscope optics, which are the key to everything because they are the optics that replace a standard condenser on a research grade optical microscope that allow you the ability to observe the scattering cross section of nanoscale particulates and nanoscale elements in a wide range of environments, such as the tissue that we looked at. And in a nutshell, what it does is it creates a highly structured oblique or shallow angle of illumination, which is a traditional darkfield process, such that this highly oblique broadband light interacts with the sample. But the light itself has a numerical aperture that causes it to bypass the objective because the objective is set with an NA so that it is below the numerical aperture of the light source, so that scatter from the sample goes into the objective and nothing else. So it would be the scatter from the tissue in the example that I showed you earlier, but also the scatter of the nanoscale elements. And you can see those very clearly and very distinctly because you have a very high signal to noise ratio. The signal of the sample scatter versus the noise or the lack of noise that's generated from the source light. And source lights can be different depending on different applications, but generally they're a very common halogen light source, which produces very smooth illumination. Just to show you an example of this signal to noise, this is a paper that was published a number of years ago by a group in Korea that used the enhanced darkfield optics for microchip electrophoresis work. And what the group there needed to do was the ability to track these iron oxide nanoparticles moving in this microchip electrophoresis environment. And using the CytoViva enhanced darkfield optics, they got a very clear signal of these very small metal oxide materials in this environment. And they also compared the performance of the enhanced darkfield optics from CytoViva versus conventional darkfield optics. And you can see a very clear difference just qualitatively in the signal to noise ratio. But when they measured that in a highly quantitative manner, they measured about a 10x increase in the signal to noise improvement. And what this means from a practical perspective as it relates to CytoViva is that you have the ability to observe engineered nanoscale materials, metal oxides, polymers such as these plastics and such, well below 50 nanometers, metal oxides and noble metal materials down to 10, 20 nanometers. In fact, you can actually see the scatter. That's been recorded in a number of peer-reviewed publications where researchers have identified those materials of that size using cross-correlative high-resolution methods. And the good part about this technique is it allows you to image these samples without any sample prep very quickly and very easily. You can capture these images in seconds from the time that you place them on the slide, on the microscope, et cetera. Capturing these high signal to noise optical images requires that you then want to be able to characterize and analyze and map what you're seeing with your eyes. And so hyperspectral imaging does that by recording the optical spectral response in every pixel of a high resolution image, okay? And so it works by capturing either the visible near infrared or shortwave infrared signals in every pixel of the image. And in the Wiener range, we're capturing data from 400 nanometers early in the visible, all the way out to the near infrared at 1,000 nanometers, or in the shortwave infrared range, 900 to 1,700 nanometers. And that data is captured in every single pixel. And pixels can be very small. They can be right at 0.1, 0.12 microns with a 100x objective, for example. And in each pixel, you've got, for example, all of the Wiener data from 400 to 1,000 nanometers. And that spectral data is very high resolution, down to 2 nanometers or below 2 nanometers of spectral resolution, so that you can query this pixel level data at very high spectral resolution. But best of all, it's presented as an RGB image so that you get high spatial context to this high spectral resolution data. And that allows you the ability to perform detailed quantitative analysis, to understand the difference, for example, between what might be a polymer nanoparticle in the tissue sample versus some other non-polymer element that may be in the tissue sample that you're looking at, for example. I want to very briefly, before I turn it over to Phoebe today, just go through two really quick examples of how nanoplastics applications are a good fit for the technique. And the first one I want is to look at identifying plastics in whole animal organisms. And this is work that was done by Professor Raoul Fakhroulen at Kazan University. And Dr. Fakhroulen is a longtime user of this technology and has published numerous methods papers on a wide range of nanoscale elements in a wide range of biological and material-based environments using the technique. And he was one of the early publishers of a methods paper with this technique. And in this case, he was identifying plastics in whole animal organisms and C. elegans. And the first thing that Dr. Fakhroulen attempted to do was to try to look at a wide range, in this case, these 3 different polymers in solution. And what he wanted to try to do was to see what differences at the spectral level he could discern between these 3 different polymer materials. And he was able to, as you can see from the spectral response characteristics that we're measuring here, to detect an optical spectral difference between these different types of polymer materials, which was a very interesting result. And you see these different polymer materials just isolated in solution. And then you see them here as well. And in this third example that we show here, these 3 different polymers have been mixed. And then he spectrally mapped each one of the 3 different polymers in different concentrations here. And so one of the polymers is mapped in red, another is mapped in green, and another is mapped in the cyan, to demonstrate the ability to identify these polymers when they're mixed together in an environment, to distinguish one from the other and know what you had. And that was very important because in the next step of his experiment, he wanted to be able to expose the C. elegans to these different polymers and understand as these C. elegans ingested these polymers, where you would expect to find them. And of course, he expected to see them in the gut section of the C. elegans, and that's exactly where they showed up. And so in this example image that we're looking at here, we're looking at A through D is one of the polymers, and then E was a different polymer, and then F was again a different polymer. And in the bottom image here, we're looking at where they spectrally map those polymers in the gut section of the C. elegans. And you can see here in this last image that we're looking at here, that you can actually see these materials, and they have a certain geometry inside the C. elegans. So there's very distinct levels of detection and discernment that were able to be determined using the instrument in this regard. And in this last image of this example research that I'm going to show very briefly, C. elegans were exposed to two different polymers, 80% polystyrene and then 80% of the PMMA, or excuse me, 20% of the PMMA sample. And then using the spectral response measurements of these two different polymer samples, mapping of the two was conducted inside the C. elegans. And you see here that most of the spectral mapping that occurs is green, which is consistent with the polystyrene. And then you see some areas of red that map, which are consistent with the PMMA polymer. And this spectral mapping and the class distribution from this spectral mapping matched almost identically the percentage of exposure that was given to the C. elegans, or that the C. elegans was exposed to, demonstrating the accuracy of the instrument to discern between the two different polymers that had been ingested by the C. elegans in this case. So a really interesting example. And then this final example that I'm going to show from an application is the identifying plastics in H&E stained tissue. And this is some of the early work that Dr. Stapleton was involved in. And so I just want to show it because it's very, very interesting and convincing work. And this is ex vivo tissue of a mouse liver. And as we know, the liver is sort of the garbage disposal, in effect, of a wide range of things that the mouse may have been exposed to. This is H&E stained tissue, but this is a negative control. And it's interesting to note that in this H&E stained tissue, you've got white particulates that are there. But we know that the mouse was not purposely exposed to any of in this case, the polystyrene, but you have these particulates there. And so with the hyperspectral microscope analysis capability, you have the ability to look at positive controls, in this case, mice exposed to polystyrene versus negative controls, mice that are not exposed to polystyrene, and to create a spectral library of the unique elements of the polystyrene exposure. And so I want to show that and show how you can then map the polystyrene without mapping other particulates that may be present. This is the Darkfield hyperspectral image of a mouse and of ex vivo liver tissue, H&E stained, exposed to the polystyrene. And we have a spectral response here, just an example. And this is a spectral response characteristic of the polystyrene that's unique to the mice that were exposed to polystyrene and doesn't exist at all in any of the negative control mice, even though those mice do have white particulates that are present in the liver tissue. And in this example, we show spectral mapping, again, of just the polystyrene spectral response characteristics. And you can see that most of the white particulates actually map, but not all of them do. And if I point out some that don't map here, we see an area here that doesn't map. We see an area here that doesn't map, an area here that doesn't map, right here. And so it can discriminate between the polystyrene and between the other particulates that are present in this case in the liver tissue of the exposed mouse to demonstrate these differences. And now as a final, we're going to show you what the spectral response characteristic was of some of these other particulates in the control stained tissue versus the spectral response of what was identified as the polystyrene in the exposed tissue. And you can see now how it's easy to distinguish between those two because they have very different spectral response characteristics when we look at the mean difference here. And so that's just an example of how the instrument works. And with that, I'm going to stop my presentation and I'm going to turn it over to Dr. Stapleton and allow her to share her data. And at the end, we will ask questions. Dr. Stapleton.
Beautiful. Thank you, Byron. Thank you so much. Thank you for the invitation and all of those things. Oh, I need your end. I can't share my screen at the moment. Can you look, Byron, on your end and see? I have the button, but it's been disabled. Just 1 second on that. I thought as a participant it would be all right, but not yet. You're all right, you're on mute, but I could lip read that last part, that's for sure. Nope. Nope, you're on mute, Byron, sorry.
I'm going to make you a co-host, which should allow you to share.
Good, let's try that. There we are, beautiful.
Okay.
Thank you.
Perfect.
All right, sorry for that. Pause, everybody. All right, there we are. So thank you again for the invitation today to share some of our work. So as Byron was indicating, I'll be talking about our nanoplastics project, our imaging and analysis using enhanced Darkfield hyperspectral microscopy, and really how that technology is fitting into our overall research plan and how we've been using it most recently. So for today, we're going to have a brief outline. I'm going to talk about particulate matter exposure during pregnancy. That's really how we kind of got into this field to begin with. We'll talk about micro and nanoplastics and translocation during gestation or pregnancy. And then we'll talk about our laboratory model for micro and nanoplastic exposures and some of our current and future directions. So we really got into this area and this question because it had been identified previously that air pollution and particulate matter, or PM, had some unique outcomes in epidemiology studies. So in theory, something you inhale would likely affect the pulmonary system. That would be the best guess for most of us of what would increase ER visits in particular. But those epidemiological studies actually indicated evidence of cardiovascular disease outcomes or cardiovascular effects after these high air pollution days, and not so much the pulmonary effects. At least these weren't the things that were creating myocardial infarction, stroke, angina. These weren't increasing morbidity and mortality rates. It was the cardiovascular outcomes. And when we continue to look at those epidemiology studies, we identify that there's also an increased risk of intrauterine growth restriction and or fetal growth restriction from exposure of the mom during pregnancy as well. And so overall, we could identify that exposure to particulate matter, both fine, which is a specific size range, and ultrafine, which is even smaller, and that's in the nano range, of this particulate air pollution has adverse effects on cardiopulmonary health. And so looking at some of that literature, we could identify the pulmonary exposure to fine particulate. These are 2.5 micron or larger. And the body has a lot of systems to be able to clear particles of that size range. So it gets caught in the mucosocilatory escalator and it gets cleared from the upper airways of our lung. Unfortunately, those smaller particles, which are ultrafine or in the nano size range, these are particles less than 100 nanometers in a single dimension. Those particles can bypass those clearance mechanisms, and they can actually get trapped in the lower portions of the lung and in those alveolar regions. So there's a greater pulmonary retention of these ultrafine or nano-sized particles in the lung. It was theorized then that this connection between pulmonary exposure and cardiovascular outcomes may be associated with pulmonary inflammation that leads to a systemic inflammation following exposure, kind of a spillover effect. It may have to do with sympathetic neural activation and the connection between the lung and the brain and the cardiovascular system. And lastly, the last theory was particle translocation from the lung to systemic organs. And we get a local and direct effect from this particle actually leaving the lung, or particles leaving the lung, and having these effects on downstream tissues. And so we began to think of this concept in a maternal fetal model. And at the time, this Time magazine, sorry for the pun, but at the moment, this Time magazine article had come out about how the first 9 months shapes the rest of your life. And a lot of conversations about the Barker hypothesis of how things that one might be exposed to during gestation and during early life would lead to an increase in risk factors of metabolic disease, cardiovascular disease during the normal life, before somebody eats McDonald's every other day, things of that nature, before one becomes a smoker. They might actually be predisposed to some cardiovascular and metabolic outcomes because of these exposures during pregnancy. We realize that pregnancy itself is just a complex and widely understudied model and lifespan because of many ethical concerns. But in that time, there has to be a rapid and precise development of tissue, including the placenta, the enlargement of the uterus, and of course, fetal development itself. In that span, there's also a great deal of hormonal variation from the non-pregnant state. And in humans, there are many physiological disparities from that non-pregnant state or normal state as well. There's an increase in blood volume, there's an increase in heart size, and interestingly, for pulmonary exposures, there's an increase in tidal volume as well. So a woman who's pregnant doesn't take more breaths per minute, but the breath that she takes is deeper per minute. And each of these components will modify the pharmacokinetics of any type of exposure, whether that's a drug exposure or in our case, looking at a toxic exposure as well. So we could see that we would get this increased likelihood of fetal growth restriction, and fetal growth restriction itself increases the risk of fetal or offspring morbidity and mortality in the perinatal time right around birth. And it also increases the risk of cardiovascular, metabolic, and neurological disease for that offspring in that F1 generation. However it's living its life, it already has this predisposition to some of these diseases. And so our research question for our lab overall is really how something a mother inhales during pregnancy, how does this affect her health, the fetal health and fetal development, and the surviving offspring as well. So we created a laboratory model and an experimental design, primarily starting with titanium dioxide. This is a very low reactive metal, and we were using that as a surrogate for air pollution overall. So all of the different size ranges, all of the different components of air pollution that we might be aware of. Excuse me. And so we have our, we're using Sprague Dawley rats currently. We would put our rats into our inhalation facility. They would have a whole body inhalation. We could put comparison control animals in our inhalation facility as well, and just have filtered air flowing through there. So the only difference between this with stress and anxiety as it pertains to that is really the titanium dioxide particles. We could keep a consistent concentration of particles for over 4 hours if we wanted to in this inhalation facility. And we have all of the characterization components to be able to identify the size range of our aerosols. And in our lab, we have them to NIST standards, which is the National Institute of Standards and Technology. Think technology, don't quote me on that part, but it's up to NIST standards. And so in that, we're able to say that we are exposing to ultrafine particles, and that's really important when you start to talk about nano-sized particles to make sure you're in that very small size range that can get all the way into the depths of the lung. And so we started with these exposures, and we identified many different outcomes using our titanium dioxide as our surrogate. We identified maternal outcomes that include uterine endothelial dysfunction. This means that the vasculature of the uterus isn't responding in the same way that it does in the control animals. That's primarily endothelial dysfunction or endothelial driven. The endothelial layer is the last layer of the vasculature. It's a single cell layer that lines all of the blood vessels, and it communicates well with the vascular smooth muscle that can then dilate or relax if you're a vascular smooth muscle, and you also constrict, which would make the vessel smaller as well, which would limit blood flow to that area. And overall, this culminates in an inability to meet perfusion demands. When the fetus gave the signal or the placenta had a metabolic signal that it needed more blood, these vessels weren't responding the same way that the controls did. So we identified many different maternal outcomes. We also looked at our fetal tissues and our offspring, and we did identify the development of fetal growth restriction, or an IUGR phenotype, and that led to an impairment of fetal cardiovascular health, and that lasted into adulthood in the surviving offspring as well. So we were able to show cardiovascular disease outcomes all the way out to 12 months of age, actually, after this exposure. So we took a step back and started to wonder about that middle ground, the placenta, which is, if maternal fetal are not studied well, then the placenta is a whole new world. So we were wondering about the outcomes associated with particle translocation specifically, and if the outcomes we're seeing in our fetus and in our offspring, are those associated with particle translocation, or are those associated with secondary effects because of the maternal exposure? Are we talking about inflammation getting across the placenta? Are we talking about oxidative stress concerns? Or are we talking about those in addition to this particle translocation? So we really wanted to know if the placenta would act as a biological barrier, or if nano-sized particles could translocate across that placenta. So we developed a placental perfusion technique to be able to identify if they really were acting as a barrier, and really wanted to know how quickly those nanoparticles could cross the barrier if they were able to. And that time would give us some indication of how those particles were getting across, if this is a simple diffusion, or if this is something that requires receptors to be able to get across. And that time would give us a little more indication of how these particles are crossing. So we developed this placental perfusion technique. In this case, we cannulate the uterine artery, both on the proximal and the distal end. We also cannulate the umbilical artery and the umbilical vein. And we could inject, if you can see it on the right-hand side, in this case, we injected Evans blue dye, which is an albumin-bound dye, so a protein-bound dye. And we put that into the uterine artery at 80 millimeters of mercury. And you can see that it's only blue on that one left-hand side. It's not blue on the right-hand side, and it's not blue as you cross the placenta into the umbilical artery and vein. And so what that indicates to us is it really does stay bound to that albumin. It's not passaging through. So we were able to get fluid passing, but that fluid wasn't blue. It stayed on the side that it was supposed to. And so that was really interesting to us because it shows that our placental perfusion technique was working. So we moved forward to add a bolus of 20 nanometer size polystyrene nanomaterials or nanoparticles. And so we did a single bolus. We didn't infuse. We just put I think about 500 microliters, something like that, into that uterine artery. We collected the effluent on the distal side, and we also collected the effluent coming from the umbilical vein. So that's in that fetal compartment that would be going directly into the fetal liver would be the next stop. So we cannulated, we injected, we started to collect this effluent for analysis. These polystyrene particles were rhodamine labeled, so we could use a simple plate reader to be able to look for fluorescence, which would indicate a concentration of polystyrene in that effluent fluid. We were able to find it in the uterine artery on the top graph here. We were able to find it within 10 minutes. It just crossed right across that placenta, right into our effluent collection within 10 minutes. And it maintained that collection and kind of went down, but maintained that collection for about 90 minutes to get all of that polystyrene across the uterine artery. And what didn't go across the uterine artery went down through the arcuate circulation, down into the placenta, and out through the umbilical vein. And we were able to start collecting polystyrene particles within 90 minutes coming through that umbilical vein. And so from 90 minutes on through 180 minutes, we stopped our exposure we stopped our collection at that point. And we were able to identify that we do see particle translocation, so 20 nanometer sized particles do get across the placenta. They do enter the fetal circulation through the umbilical vein. So we identified that the placenta does not act as a barrier to nano-sized particles. And I gave this presentation at one of our center meetings at Rutgers, and as we were leaving, I was very relieved that my talk was over and I had answered all of my questions well, and met a colleague at the elevator who is a senior in comparison to myself at the time. And he said, well, I'd like to invite you to this microplastics seminar. And I said, but I don't do microplastics. I do air pollution. I do titanium dioxide. I thought we just went over all of this. Clearly, I didn't explain myself well. And he said, well, you know that polystyrene is a plastic, right? So you're doing plastic exposure. And I realized, oh, I am. I was doing nanomaterial research, but in fact, I'm actually doing plastics. And once somebody says that to you, it's a change. So I apologize if you haven't had that change in your life yet, because you will after this conversation. But we are surrounded by plastics. We are inundated by plastics. They are legitimately everywhere you look, at the computer that we're talking over today, to the container that you likely had coffee in this morning, but we're just surrounded by them. And from 2025 to 2050, it's expected that increase in production of plastic will increase by about 30% in the next 25 years. So it's increased exponentially from 1950. It shows no signs of slowing down, only other uses for these polymer-based products. And so we have a significant amount being produced now, and that's only going to increase in the near future. So once you look at how much plastic interaction we're having on a daily basis, you start to consider the life cycle of these plastic products as well. So plastic is formed from oil, and so it's extracted from those raw materials. The production is a chemical production where the monomers from the oil is put together and chemically linked to make polymers. Depending on the type of monomer you use and the type of chemistry that's included or plasticizing chemicals that are included, you get this manufacturing of a polymer or a plastic resin. From that, there's fabrication of the variety of different products that you might be producing. This can be a hard polystyrene, a very flexible nylon that is used in clothing or camping equipment and things of that nature. You have this end product, whether that's a tire, a cell phone, a blanket. It can be used in packaging and Amazon deliveries. It can be used in distribution. So we have the use and reuse of some of these products. And then we get to the end of life. Only about 8 to 10% of plastics are recycled, depending on where you're located. And the rest of those go to landfills or energy recovery centers. And energy recovery centers are a really kind way to say that we incinerate them or that we burn them. And so we're going to the 90% will be going to landfills or going to these energy recovery incineration plants. So in that recycling, landfill, energy recovery, it's been estimated that the breakdown of plastics can range between 10 to 1,000 years. And that has to do with thermal and UV and mechanical and chemical degradation as well. And the average estimates for a plastic bag, so just a single-use plastic bag that you may be able to get at the grocery store, those plastic bags can take up to 20 years to degrade, and a plastic bottle breakdown is estimated to be at about 450 years. And I keep saying the word estimate because no one's really tracked how long a plastic bottle takes to break down over a normal real-world experience because it hasn't been in existence for 450 years to track that, nor has anyone been here to watch it. So it's an estimate of how long it's going to take for this to break down. And it's also an estimate or a gray area of what one means by degradation. So this concept of decomposition or degradation is really unclear. It's not always referring to that chemical breakdown of taking a polymer and breaking it back down to its single monomer form. More often, it's referring to particle size in the idea that if we can't see it, then it must be gone. And the concern with that is that those particles that we can no longer see end up in the environment then. And for many years, laboratories, including my own and others that I had been working in, we weren't even tracking these in the environment. When we did air pollution studies and we had our filters and we collected all of that air pollution, we were using techniques like ICP-MS to be able to get to that ionic base of what those chemicals were in our powder, in our dust. And polymers weren't even included in those analyses. ICP-MS is not an appropriate way to be able to measure polymers because of their organic structure. So we were overlooking the fact that they have been in our environment for so long. Now that we're aware, we're finding them everywhere we look. We're finding them, in this case, looking at the metric tons of plastics that are able to be identified within our national park system in the wilderness. So this is a range of national parks and wilderness areas, primarily out west. But you can see just the amount of particles and micro-nanoplastics that are being able to be identified in our environment. Even more so, microplastics and likely nanoplastics are also in our indoor environment. They've been identified as within our food sources, within our beverages, so we know we're ingesting these particles, looking at bottled water, beer, wine, tea, rice, sugar, and honey, so things that should be considered organic, like honey. And we know we're bringing them in because we've been able to find them in fecal matter as well. So we know that they must be going in if we're able to find them as they're coming out. They've also been identified in indoor air, so we know that we're inhaling these particles. It's been identified that it's about 100 times more in indoor air than outdoor air. And they microplastics have been identified in human lung samples, both of patients undergoing surgeries and cadaver samples as well. And so this is especially concerning considering how much time humans are spending inside, especially post-COVID time. But in 1979, we were already spending 70 to 90% of our time indoors, and during COVID, that only increased. And so that indoor air is also of concern. So as a definition, micro and nanoplastics. Microplastics are not a specific type of plastic. It's not necessarily only polystyrene, for example, but it's any type of plastic that is a fragment that's less than 5 millimeters in length. So for a mental image, this is a sesame seed, for example, or on the toothbrush, the bristles of that, those bristles in width are less than 5 millimeters. So it's safe to say everybody was exposed intentionally to microplastics this morning if you brush your teeth. Nanoplastics then are defined as two different ways depending on who's talking about them. So for environmental studies, they're identified as less than 1,000 nanometers or less than 1 micron. It's considered a nanoplastic. And in laboratory studies like my own, they're identified as less than 100 nanometers in one dimension. And that's because many of us are coming from that nano and ultrafine particle size range. So that's how we've been communicating about these nano-sized particles for well over 20 years at this point. And so 100 nanometers is laboratory studies and less than 1 micron is environmental. And that puts these in the size range of a virus, of an antibody, puts us in the size range smaller than a bacteria. And so also using that COVID idea, we know how hard it was to try to avoid that coronavirus, and these particles are well within that same size range, if not smaller than some of the coronaviral variants that we see. So with nanoplastic exposures, first a consideration, because those nanoplastics are at the very least 1,000 times smaller than the average microplastic particle. They're extremely difficult to identify outside of a laboratory setting, and Byron connected to some of that when he was speaking earlier in that mixture idea that outside of a laboratory setting in a real world environment, we're not exposed to a single plastic. We're likely exposed to a mixture of things. And so that makes them even more difficult to identify when we don't even know what it is that we're looking for, what kind we're identifying. And because of that organic chemistry that they have, those polymers are very difficult to assess using analytical techniques. So the three primary techniques that have been used at this point are the hyperspectral darkfield microscopy and that light refraction, as Byron was indicating before. Pyrolysis and gas chromatography, mass spec, has also been used. But please keep in mind that pyrolysis and gas chromatography will identify the polymer. So they're burning the plastic, and that's not really identifying the particle itself, but it is identifying that polymer is within the tissue or within the substrate that they're using. So it's identifying that the plastic is there, but not necessarily the particle component of that plastic. And then lastly, Raman spectroscopy is being used, and this uses lasers to be able to identify and stimulate the polymer that are there as well. And the microscopy component of that can then identify the size of that particle once the polymer has been identified of what it is. And we have used that Raman, specifically the stimulated Raman scattering microscopy, and we've used that to identify nanoplastics in bottled water. In January, this paper came out. And we were able to identify in our collaborative group that there were about 240 to 400,000 nanoplastic particles per liter of bottled water. So we were able to show the microplastic side with this technique, but then we were also able to show the nano-sized plastic of this technique as well. So the great thing is we're able to identify some of those mixtures, especially of unknowns. But the negative side is that we're only able to identify plastic particles that are about 200 nanometers or larger. And so that's right within that fine to ultrafine range. We're still from a laboratory setting, in that microplastic size range at that 200 nanometer. So while this technique is up and coming, we're not able to identify that nano range at this point. So we really are interested in that nanoplastic identification. These are going to be the plastics and the particles that are most likely to get across those epithelial barriers. And so we turned to the hyperspectral darkfield microscopy, and specifically CytoViva, because we'd used it in the past. We were able to identify multi-wall carbon nanotubes after exposures in our past from some of our occupational studies. And as Byron was alluding to, this is important because we're able to identify the polymer as being something different. I refer to it as looking at that light that's been refracted back from hitting the particle. It's different than that hits at a cell. And so the computer system is able to identify what is a cell, what is a particle. And not only that, we're able to do it on a pixel by pixel basis. And so we're able to get well into that nano range of our particles to be able to see what has translocated, in our case from the lung, but we've used other tissues as well. And so we realized that the placenta does not act as a barrier to nanoplastics in addition to nano-sized particles. And so we really wanted to understand the whole system then. If we're able to put these particles within the maternal system, not just into the uterine artery, but into her lung, into her GI system, will we be able to find them in these fetal tissues then? So we set up an experiment. So there's a lot of math associated with that, a lot of dosimetry. We wanted to make sure that we were doing something that was real world relevant. So we used a paper coming from Cox Group out of 2019 that had estimates of how many particles one would be exposed to. Using that, we did some additional math to translate down from microplastic exposure, which is what their group had estimated, down to nanoplastic exposure. So happy to talk about all this math, but also happy to skip right through it as well, because that's not really the area of interest. And so we did the dosimetry as well to get from the human to the rat. So we really wanted to check off all of our bases, that we were using nano-sized particles within a real-world exposure amount, and that it would be relevant to be able to translate or estimate from a human down to a rat. So we'd be able to go back. And what we identified is that the experimental dosage that we were using and we were approved to use was actually lower than the calculated environmental dose for a human woman, of course, but during pregnancy, with that increase of tidal volume as well. So we're well within our environmental exposure dose. We put these micro nanoplastics into the pregnant rat. Those particles would have to, in many cases, for our pulmonary exposure, they'd have to leave the lungs, they'd have to go to the heart, they'd have to go down the uterine artery, they'd have to cross the placenta. From the placenta, they get into the umbilical vein. From the umbilical vein, they actually go right into the ascending inferior vena cava, but also a little bit hits the liver first as well. And so they go right into that fetal circulation. So the rat that's here on the bottom left, highlights the tissues that we looked at from the rat. So starting with that liver and then the other systemic tissues, again, from that fetal time point. So we did these exposures. We put the particles in the lung through intratracheal instillation. We left the animal for 24 hours after exposure. We came back, we did our dissections, we took our liver, we stained them with H&E stains so that we could have a pathologist view to see if there were any gross morphometric changes or histopathology changes. And we also worked with Byron and his group at CytoViva to be able to identify these nano-sized 20 nanometer, rhodamine labeled polystyrene beads in our fetal livers within 24 hours of exposure. And the white arrows are pointing those out. Not only did we find them in our fetal liver, we were able to identify them in our fetal lung. Again, these were through maternal exposures. These are still fetal tissues 24 hours after exposure. We exposed at gestational day 19 and sacrificed at gestational day 20. So rats usually deliver between gestational day 21 and 22. So these are fully formed animals at this point. We found them in the fetal liver, the fetal lung. We found them in the fetal kidney, the fetal heart, and the fetal brain as well, which was a really interesting finding on our end. We repeated these studies using a gavage technique instead. So instead of putting them into the maternal lung, we put them in the maternal GI system and did the same thing, actually directly into the stomach, and did the same thing. We waited for 24 hours. After exposure, we sacrificed our animals. We were able to identify these particles, again, using the hyperspectral microscopy. We were able to identify them in the rodent placenta, in the heart, in the kidney, and in the liver of the fetal pups after gavage. And that's not to say that we didn't find them in the other tissues. That's to say that we didn't assess them in the other tissues. So that's a point to keep in mind as well. So from that, we came back out to our laboratory model and our experimental design. And in this case, we were forcing the particles into the lung and into the stomach of these rodents. And in our inhalation world, inhalation really is the gold standard to be able to have these exposures. There's a lot of stress. That stress can change the physiology. So if we can have a more natural inhalation for micronanoplastics, that would really be a better avenue for us to do some of our assessments. So in this case, we developed a micronanoplastic inhalation technique and methodology. So we purchased commercially available polyamide. This is a nylon particle, bulk microplastics. It's a food grade nylon at that. And we have a set point of 10 milligrams per meter cubed, which is a industrial hygiene nuisance dust dosage, but it's an occupationally relevant dose at the same time. So we aerosolize our particles here. So they use an acoustic generator to be able to aerosolize them and put them in the air. There are many different components that are measured in real time to be able to understand the dose, the temperature, the humidity within our inhalation chamber. And the animals sit in our inhalation facility, they fall asleep, and they breathe in these particles. We were able to characterize them with our techniques. So the commercial characterization identified these particles as having a diameter of 5 microns, or with an error of about 1. So this is well within that microparticle range. And when we aerosolize those, the smallest particles get lifted into the air. And what we identified using both a 1 milligram or a 10 milligram per meter cube dose is we do get a very large nano fraction that's in that aerosol dust as well. So we get many, many particles that are within that 100 nanometer range. And that's measured in a variety of different ways, whether that is electromobility that's used in our SMPS or APS technology, or aerodynamic mean as well. So thinking of a javelin can be very, very long in one dimension and very, very narrow as the person's throwing it in the other dimension. And so that's what that aerodynamic diameter is, that very narrow range. And so we feel that this particle and that this characterization is reasonable for inhalation toxicity and toxicology measures looking at micronanoplastics. We did a study where we looked at the systemic outcomes. This is a single inhalation exposure, so that same 24-hour idea, this time using our polyamide particles. In this case, we were using non-pregnant female rats. We did take care to make sure that all of our rats were in the same stage of their estrus cycle, and that stage was estrus itself. So this is the most fertile time point of the rat cycle. We didn't see great pulmonary inflammation or great pulmonary toxicity outcomes. We saw no histopath concerns. We did a BAL, a bronchoalveolar lavage, to look at the cells within the pulmonary fluid, and really didn't see a huge shift or change. We saw some neutrophils in our exposed animals, which we did not see in our control. But the purpose of this study was really to get a wide toxicological outcome of this single inhalation exposure. What systems are we looking at, or should we be looking at more closely? So we looked at the pulmonary system, we looked at the cardiovascular system, both the microvasculature and the macrovasculature. We did see some endothelial dependent differences and even endothelial independent differences in our uterine artery, in our radial artery, as we get closer to that fetal area or to the uterine horn itself. So we did see some vascular outcomes despite not seeing any pulmonary outcomes. Further, we did see some systemic inflammation. So this was in the plasma of these animals, again, 24 hours after exposure. We saw an increase in IL-6. We saw an increase that didn't meet full statistical criteria, but it was less than P 0.1, but not less than P 0.05. But we did see increases in IL-6, C-reactive protein, and MCP1. We also evaluated the endocrine system, and that was probably the most interesting component for my end, because polyamide is not known to have a plasticizing chemical. It isn't known to use phthalates or BPA or things of that nature that are known endocrine disruptors. It actually uses water as its plasticizing agent to change the flexibility. But even in that case, we found that there was a significant reduction in 17-beta estradiol, which is an estrogen mimetic that's necessary during pregnancy. And again, this is the most fertile time point. So we do have some questions about fertility after this micronanoplastic exposure. We've moved that into our pregnancy model, and in those characterizations, we're able to see that we do get a fetal growth restriction phenotype after repeated exposure to our polyamide particles from early pregnancy, gestational day 5, all the way through gestational day 19. And these animals are sacrificed on gestational day 20. We see an increase in placental weight, which doesn't really correlate well with that fetal weight decrease. So that's a unique outcome for micronanoplastic exposure. We also see a reduction in the number of fetal pups, but that's not to significance, and it's likely associated with a significant increase in fetal reabsorption, and that's a miscarriage in a rodent model. So we do see some pregnancy outcomes. We wanted to know if these particles stayed in the fetus, though. That was one of our major questions, to be able to identify or understand that F1 generation increase in or change in health if exposure to micronanoplastics during pregnancy, could that be considered a risk factor for other disease states in the future? Also, do those particles then bioaccumulate? We know that they're getting in. We're a little unclear of how they're getting out at the moment, though, if they're getting out in those same vast amounts through urine, through feces. So we wanted to know if those particles stay with those fetal pups after delivery. So we put this study together that the group at CytoViva graciously helped us with. And in this case, we did the micronanoplastic exposure. These animals, the mothers, inhaled particles from gestational day 5 through gestational day 19. We left them alone at gestational day 20. We let them nest and deliver their pups. We let them nurse their pups up until 2 weeks of age, and that's the point that we sacrificed them. And we looked at those tissues, and we were able to find our polyamide in particular. So we weren't really worried about cross-contamination of what other plastics the animals might be exposed to during that time, because our polyamide is so unique in its wavelength and characteristics. It's not used in our laboratory for any other nylon, and this polyamide 12 in particular are not used in our laboratory for any other outcome. So we're very confident that this was a exposure-driven outcome and not a cross-contamination outcome. We were able to find those particles in our offspring liver, which is here in A on the green one in the upper left-hand side. We're able to find them in the lung. We're able to find them in our offspring kidney, many in the heart, and many in the brain as well, which is an interesting outcome. And so I wanted to understand how long they last there. So we also have looked at those same exposures, but we looked at the progeny at 3 months of age, and we've been able to identify those polyamide particles in our tissues at 3 months of age, both male and female rats, in the liver, in the heart, and in the brain as well. And again, that's not to say that they aren't in the other tissues. That's to say that we haven't finished looking at our 3-month tissues, nor have we finished looking at our 6-month tissues either. So we want to understand how long these particles can persist in our F1 generation. And again, they were only exposed during pregnancy and only exposed during gestation. So these animals have not entered our inhalation facility. So at this point, we're going to continue our micronanoplastic studies, and our directions include the following. We really want to understand the mechanisms of that micronanoplastic uptake and that micronanoplastic translocation to those other tissues. We want to understand if those particles are in the interstitial space, are they just in the tissue, or have they really been taken up into some of those systemic cells? So are they in the hepatocytes of the liver, or are they in the interstitial surrounding, or they remain in the vasculature? So really understand where they are to understand what the toxicity could be. We want to look at those systemic toxicities of the micronanoplastic exposure and, of course, the deposition. We want to understand the specific toxicities based on the physicochemical properties of the particles. So different polymers are going to have different toxicities. Also, the life that those plastics had, not just the naive ones, but maybe the ones that have been incinerated or the ones that were near a landfill, how that changes the polymer, but also how that changes the toxicity of the polymer. We want to understand the internal dosing and bioaccumulation. We know that they're staying in these animals in the F1 generation. Can we increase that internal dose then if these animals are then exposed as well? And so how does that bioaccumulation occur? Are these particles able to get back out of these animals? And then lastly, we're very interested in a comparison of those available micronanoplastic quantification methodologies. We're right on the cusp of a new group every day, it seems, finding plastics in human tissues. They've been found in everything that's been examined at this point. And many of those are using this GC pyrolysis mass spec to be able to identify that polymer in a variety of different tissues. And we really want to identify if it's the particle itself in those tissues as well. And so we'll need to compare those methodologies between the SRS Raman, the GC pyrolysis, and the hyperspectral microscopy, because each one has limitations associated with it, but each one also has huge benefits associated with it. So if we can kind of validate each of those techniques using another one, I think that'll drive the field forward quite a bit. So lastly, I just appreciate this meme greatly, that there's a little Barbie in all of us, and that's because of the microplastics. So with that, I'd like to thank my group that's here at Rutgers, and specifically those that are bolded, of course, our collaboration with CytoViva, and our collaboration with the pathology services, molecular imaging facility, and our funding sources. So thank you very much for your time at this point, and I see some questions coming in the question and answer component. So thank you very much.
Phoebe, before we get started, and we're going to go through some of these questions really quickly, you talked about, I think one of the important aspects of this is to have as many corroborating methodologies with this. And will the GC mass spec pyrolysis data, will that give you volumetric related information about the polymer's presence in the tissue?
It will. It'll give us that can give us a concentration of how much, because we know how much tissue we're grinding up, for example, and then and then burning. And so that gives us a concentration of polymer per, say, gram of tissue or whatever it is. And not only that, it can do a variety of mixtures as well. So it gives us a gram or an amount of polystyrene, an amount of polyamide, an amount of name your polymer, or almost to that point. They're still collecting data for their libraries as well, but in that gram of tissue.
Right.
The problem is we don't know if that is a full particle then, or if that is the chemical that's there at that point. And that's where the rate limiting step is.
Gotcha. Okay. Maybe we'll go through a few of the questions that we have here, and we'll see which one of us might be best for answering this. One was Gabrielle Sylvia thanking you for a great lecture. She's having some difficulty analyzing tissue sections with CytoViva, the resin that shines a lot and hinders the data collection. And she was curious about how your tissues were prepared and what kind of resins were used.
Yes, so we use, I knew that was going to be a question, and it's not on the tip of my tongue. So I'm going to have to take a minute to remember how they're prepared, because we do a lot of collaboration with the research pathology services, and they're amazing that we give them our fixed tissue. They prepare it for us and send it back over. And it is with, oh, my lab is going to be so mad. I'm surprised somebody hasn't texted in to tell me the answer. So Gabriella, I will get back to you and I will type in your answer, but that is a component of it. And part of that is we slice them at approximately 5 microns. I know that, to reduce the shine component of it. Paraffin, we fix them in paraffin. I knew it would come to me.
Yeah, they're paraffin embedded, and I think I actually think you're deparaffinizing them as well, Phoebe. And
we have not been deparaffinized.
Oh, you haven't? Okay.
No, because, and that's one of the issues that we're going to have when we start to work with the GC and with the Raman group as well. So it's really been a problem with our Raman studies, actually, the paraffin, because the lasers melt it first.
Yeah. And I would say, Gabriella, that the thickness of the tissue makes a huge difference. If you have paraffin embedded tissue that's 10 microns or 20 microns, that's a significantly different issue with regard to the scattering of that paraffin versus if you have the thinner sections that Phoebe talked about that they're using. Also, not only for Raman and for the other techniques, also for CytoViva as well, you get better results if you can deparaffinize because you remove the scatter effect of that. But we don't see a lot of scatter with Phoebe's samples, and that's from the paraffin. I thought they were deparaffinized, but it's probably just a result of the thinness of the sample, which, that the thinness, it creates some advantages, but some disadvantages. It really, I guess, technically requires that you want to use more samples if the samples themselves are thinner to try to look for the results. Here we have another one from South Africa and wants to know if there are any published guidelines specifying which plastic polymer standards should be bought and analyzed when you're conducting these analyses. I understand that assessing plastics in the lab is very different from plastics in the environment. So
you're absolutely right, Charlene. So thank you for joining us from South Africa, first of all. And there are not published guidelines at the moment to specify which plastic polymer standards should be used. So we've done as much as we can to kind of go through the literature and create a rationale of what we're using and why we're using it and how that's important. I mentioned some of that with our polyamide studies. We know that it's not used, nylon's not used in our laboratory, so we can eliminate cross-contamination risk. We use a specific nylon, which is nylon 12, which also is different than nylon 6 or polyamide 6 or polyamide 66. And so being able to kind of rationalize why you're using something, polystyrene is widely utilized, and that's because it is kind of a representative of the laboratory. It's starting to get some negatives associated with that because people have been using polystyrene beads in particular, and while they're easily available from a commercial standpoint, they're not really representative of that real world capability with fragments and different surface chemistries and things of that nature. So at the moment, there really isn't a guideline or standard. I think the European Union is working to come up with some of those components, but it's not out there yet. So the best I can suggest is making an educated choice of what it is you're going to bring into your laboratory, what it is you're going to be able to use, and then stand by and defend that choice and explain those limitations of your selection, but also the pros of what it is you selected, and explain that in your discussion of your manuscripts as you're putting them through.
Awesome. And then Dominic Lucher has a question that was along those lines, and you started to answer that, but the question from Dominic was really, if you want to do experiments with real world micro nanoparticle plastic samples, is there a chance of doing this, or is there just too less micro nanoplastics in water, air, etc.? So
it's not that there are two less. I think that they're there. I think our problem at the moment is the intersection of exposure science and biochemical physiology, toxicology science. And that's the difference of what exactly that is. So all groups are having an issue of being able to identify what exactly is in the air, what exactly is in the water, and not only from a polymer standpoint, but more importantly from a size standpoint. It's very challenging to measure ultrafine particles in the air, and then you add that you're only looking at a small fraction of them being the nano-sized plastics. That adds a challenge. So I know some groups are kind of looking at real world in occupational settings, real world in laundromats, for example, looking at the fibers that are coming off of clothing and things like that. It's just very hard to take those real world samples and be able to get a micro nanoplastic concentration of them. And then in addition, the amount that you would get would be challenging for us to do in an inhalation model if they weren't breathing in kind of everything, like road dust, for example. You get a high concentration of the tire polymers, but it wouldn't be limited just to that. So back to Charlene's answer, in that it's important to understand what it is that you're studying and also what it is that you're not studying, and therefore the limitations of the real world versus the laboratory world. So just being very clear that what we're analyzing may not be exactly real world relevant, but we understand those limitations and we're doing the best we can with what's available. I think as techniques come up, that transition to real world would be better and better.
We had a question that I'll answer, and it's related to when you're analyzing these nanoplastics in a biological media as opposed to just analyzing them on a glass microscope slide. What procedural or operational differences are required when using the CytoViva instrument? And so it gives us an opportunity to explain the process just a little bit. Hyperspectral microscopy, we consider it generally to be a comparative analysis method. Okay. And what that means is that we're comparing these polymer exposed tissues, in this case, to negative controls every time. And our development team, which was led by Jim Beach and Jamie Ertz back in the day, worked with the hyperspectral image analysis software vendor to develop an analysis technique to be able to build a spectral library of these nanomaterials in whatever environment they're in, and then filter them against that environment. Okay, because you keep your experiment design set up so that there's one variable, and that variable in this case is the polymer exposure, right? And so the environment can absolutely change the spectral response of the polymer in this case. And so, for example, polymer that's in unstained tissue will have a different spectral response than polymer that's in stained tissue, right? Certainly, if the polymer itself has a label on it, that's going to change the spectral response of it. Predominantly, it's going to be that fluorescent label. So your experiment design has to take into account capturing the spectral response of the polymer material in its environment, and then filtering that spectral library against that identical environment to identify those characteristic differences in the spectrum, and then use that for mapping. And every time you change the environment, you have to change the spectral library. Now, that can be a hassle, but it also is a real benefit because it allows you to cross-correlate that in these different environments and find it in those different environments. And so a lot of credit to our development team back in the day that understood this and created this very unique filter spectral library routine to be able to filter that spectral library against those different environments, to be able to, with confidence, identify them and map them in those environments. Then, of course, when you cross-correlate that with some of these more quantitative mass spec methods, you expect a similar correlation to that. And so we're excited about that. We have a question about, can studies of longer term effects of microplastics exposures be devised in other words, additional years?
I think that's where some researchers are going, is to understand those long-term effects, especially in our rodent models, because I think it's a day in the life of our rodents is approximately a week in the life of a human, or a month in the life of a human. And so we are starting to look at some of those longer term effects. From our standpoint, I want to understand where those plastics are first, and then be able to look at those effects of microplastic exposure. I think we're starting to see some of that in humans, especially with the study that came out from the group looking at the carotid artery plaques that came out in March. So I think some of it's going to have to be done in rodents, primarily because the only time we're able to get human tissues, with the exception of the placenta, is during times of disease, otherwise, or cadaver patients. But we're not seeing samples from individuals that are otherwise healthy. They've all come from some type of other procedure that's been taking place. And that begs questions of cross-contamination with medical devices then as well, and with saline bags and needles and syringes and all of the single-use plastics that are in that medical field. So I think, Jim, I think that's a great question. I think that's probably one of the areas that the field is going into, is to understand the bioaccumulation component, because when we look at our human samples, in order to get a true control now, we have to find samples that were collected prior to 1950 to be sure that we're actually getting a true control. And otherwise, it's just a matter of how much plastic will be in our control tissues otherwise. So I think that is going to be a component to understand the generational effect of how much plastic is in our bodies.
I know it's a really hard task. We've got a group that's looking at inhalation studies in Birmingham, Alabama, from steel mills there, and trying to use old cadaver samples to do that. And it's a very challenging experiment to try to design, and even more challenging to try to execute. So it's
It's amazing.
Yeah,
It's amazing.
absolutely. Additional question from Dr. Vermeer is how to know that commercial water bottle could have nanoplastics, i.e. after how many days of packaging of the water could it have nanoplastics?
So thank you. Yes, that's a great question as well. So for that study in particular, one of our graduate students in our collaborative group had gone to a big box store in the United States. And so they purchased 3 cases of water from 3 different manufacturers. So 3 different brands of water were purchased. And the interesting outcome that answers your question is that the plastic within each one of those brands was different, the primary plastic, and the percentages of the plastic within those brands were different as well. And so one would assume, just as you're describing, that the plastic that we found was due to the packaging or due to the capping, and maybe some heating and not freeze thaw, but sunlight, and just how long the water had been sitting in the plastic bottle would indicate how many particles that were there. And that might have been the case for one of those brands, but for other brands, the plastic particles that we identified were not necessarily from the packaging. It wasn't the same polymer that we had found. So likely that was from upstream before that water ever hit the bottle. And so we would have to do the studies to identify exactly what component of the processing led to that inclusion. But we were finding plastics like PVC, for example, which is used widely in tubing, but not in the package necessarily. And the same with polyamide, which is used a great deal in the filtering process, but again, not in the packaging as well. So those nanoplastics that we found were likely from upstream components of that commercial bottled water, in addition to some from the packaging itself. You're absolutely right. But for some cases, that wasn't the primary polymer.
An additional question was, is there a place to get real world micro nanoplastic samples? I'm not aware. I know a lot of research labs are working with engineering groups to try to create real world micro nanoplastic samples. Phoebe, do you have any thoughts on that?
I haven't seen that yet. I've seen some companies coming out with kind of packages of characterized particles.
Right.
I know some groups are working on creating a polymer library and creating just as you're describing, that real world plastic sample, similar to they have at NIST, where you can get like diesel exhaust particles that have been thoroughly characterized. I know some groups are working on getting that for micro nanoplastics as well, that characterization library. I'm not sure that they're at the point of being readily available. And so one seems to be trying to get these real world samples from the real world. And they're looking at, like I mentioned before, they're looking at dryer lens, they're looking at packaging of things. I know there was the group that had done the research on tea, for example, and had put the tea bags in the hot water and could collect the particles in that way.
Right.
So as I understand, if we're looking for real world plastic samples, then we're actually getting them from the real world, but then you have to do that characterization to show what it is that you're utilizing.
Okay. Finally, we have a question for Abner Nunes, and Abner was talking about using highly heterogeneous samples of microplastics in terms of shapes and sizes, and building quality spectral libraries from that. I'll take that one, Abner. It's I've actually worked with a group in Scandinavia that was looking at samples that were very heterogeneous, and we didn't see a lot of spectral differences in size heterogeneity. It was really more to do with the differences of the different types of polymers, but the size difference can make a difference in terms of much more scatter efficiency as the particles get bigger, and that creates some problems among those. but you might want to just use the traditional method of building the spectral library and filtering it against the different sizes to see what differences that you have there. But generally speaking, in some of the experiments we did in Denmark, different sizes and different shapes of the polymers did not create, to my recollection, material differences in the spectral response characteristics in the spectral library when we filtered them. But I'll go back and look at that, and I think I have a way to contact you, Abner. So I'll try to do that if necessary. So I think that's all the questions that we had, and we're a little past our time today. But Dr. Stapleton, thank you so much for your time. And everyone that's still on the webinar would know that this is receiving lots of press. I think when we were in Dr. Stapleton's lab a few months ago, my colleague Jamie Ertz said that, wow, said she was busier than the president with press conferences. It sounded like, as this news was just coming out about fetal transfer of nanoplastics. It had a lot of real world interest in it. So thank you very much. So.
It does. Thank you. Thank you very much for giving me I'm going to stop sharing now. Thank you very much for giving me the opportunity, though, to present our work. And so if you're still on the call, I can be reached by email. So just let me know that you were on this call and have a question if it didn't come up now. So thank you. Thank you for giving me the opportunity, because I always like talking about my science, as Jamie knows. So.
Absolutely. All right. Thank you, everyone, for attending. And please contact us at CytoViva if you have any questions that we can help you with as well. Thank you for your time. Bye-bye.
Thank you. Thanks, Darren.